this is pretty much maximum amount of stomach acid in your stomach. Is that a lot or a little in your mind? A little, right? But what if I told you your stomach is about the size of your fist? Just one. But it's empty. So how does a Big Mac, large fries, large soda fit in there? And apple pie. It expands. And so for most people, you can have your fist about this large expand up to 1 liter, 1.5 liter, and you're like, done for the day, going home, going to bed, everything's over, right? 4 liters would be your like hot dog competition eating friends. Drink. This is actually the reference I've been using all day. This is about 1 gallon of milk, and that's why the drinking 1 gallon of milk challenge makes absolutely no sense, and that's why you'll throw it all up. So for number 5, it starts off reviewing enzymes, assuming that you already got some exposure during chemistry. So we're gonna see if your chem teacher did you justice here. An enzyme acts as a catalyst, which according to our guest speaker last week, is a nice person that'll something, I don't even know what he was referencing. Anyways, is a catalyst, okay? What does a catalyst do for you? Speeds up chemical reactions without being consumed. Whoa. That means they're reusable. Good. Now the question though is how does it manage to speed up a chemical reaction? Lowering activation energy. So the example I've been using today is, let's say I start charging cover for you to get into class. It's going to cost you $50 if you want to get to class, and the only way to get an A is if you get into class. $50. And the only way you're going to make money, since you don't have time with all that homework to get a job, you have to ask your friends to borrow money, which means you're going to have 50 friends to each lend you $1, assuming that they all like you. Okay? How long do you think it'll take for you to borrow $50? A long time. What if I'm having a special though, and it's only $25 to get in today? Probably take half the amount of time, okay? And so that's what a catalyst does. It lowers the amount, the cover fee, the cover charge, and so you can get things done faster. You can get into the room faster. So it's going to lower the energy that's required by the chemical reaction. Lower the energy that's required. Good. So that should have been all from chem. We're actually going to do a lab, maybe I think next week, where we look into what affects how fast or how efficient an enzyme can get, which some of you might have done in bio already, but that's fine. So this word here, I've been getting a lot of feedback that this substrate term is confusing in relation to an enzyme. I think the book itself is a little confusing, so we'll redefine that later. But how do you name enzymes? Okay. So an enzyme's name has 2 parts: your prefix and your suffix. Based on this list, can you guess what enzymes pretty much always end in? A's. Not all. Not all of them, but some of them. Prefix, though, is usually the name of the thing that's getting broken down. Thing that's getting broken down. So, for example, amylase, the starch that amylase breaks down, though, has its own name, amylose, which is when we talked about it in the saliva, that's the one that helps break down starches. This is where it gets easier. Amylase is probably one of the harder ones. This type of enzyme breaks down a general group of macromolecules. On your whiteboards, take a guess. Proteases break down. This one is easier, right? Yes, I see a lot of proteins. You don't know how to spell protein? Just spell it out. Everything sounds like it's supposed to spell, right? Lipase, easy one, last one. Show me real quick. All right, don't erase it. I'm going to need you to dig deep, back to your bio days. Give me an example of a lipid. Whiteboard. Come on.
Thank you.
Sugar's a carb. Show your partner your best guess of the lipid. Fats, which means oils, butters, Cheetos, has lipids. If you are lactose intolerant, that means you cannot break down the sugar called lactose because you don't have an enzyme called lactase. Really simple, right? So easy. And then you talk about pepsin, and it's like, what happened? Why did you name it pepsin? Yeah.
Were those just examples, or are those like the three that we generally need to know?
These are just examples. Amylase is a very specific to our lecture. Amylase is important for us to know. The other are, yeah. How do you make enzymes? Okay, this is where we dig really deep. Way back. Enzymes. I don't know why I abbreviate enzymes easy, but I do. An enzyme is technically just a protein. What do you call a process of making a protein? Protein synthesis. Later in the unit, we're going to get really specific about the steps of protein synthesis. But in general, you have to have directions on how to make these enzymes or how to make these proteins. What are the directions of life called? DNA. Where will you find your DNA? In your cell. In the nucleus of the cell. However, unfortunately, the DNA is too large to leave the nucleus with its information, so you're going to have to make something that brings that information out. RNA, and do your bio teacher proud. What kind of RNA? M, the messenger. Remember, it brings the message out. mRNA, okay. And then, of course, we leave the nucleus. And then where do you go to actually make the protein? It's okay. We'll get back into it next week. This is just a really quick review to the ribosome. Okay? And it's once you've made it in the ribosome, these enzymes can either go outside of the cell to do their work or stay inside of the cell to do their job. It'll depend on what their job is. But basically, there are two types of enzymes. Enzymes that help break down things or help build molecules. Okay? So you've probably heard of the word metabolism before. Metabolism is basically these two reactions or types of reactions combined. I'm probably going to have you add that somewhere to the side. Equal to your anabolic reactions plus your catabolic reactions. So let's say you ate a really, really big steak for lunch. I don't know where you got the steak from, but there's your steak. Okay. You eat it, mechanical digestion, chemical digestion. You break it up into smaller pieces as it goes through the digestive system. You start absorbing those things that are broken up, mostly protein. And then why would your body want protein? What would it build with that? Muscles. You want to build muscles. In your cell membrane, there are structures that require proteins. You want to make more protein pumps. And there's a bunch of other things you want to start making with that protein. So basically, the way they define catabolic reactions is when you're tearing things up. Think cannibalism, you're like ripping someone apart. I know, sorry, too extreme. And then anabolism is where you put pieces together to form something else. Okay? So I know some of your drawings were slightly confusing. You do like little puzzle piece looking things. So if you want to write a brief description for your puzzle piece drawing, you might want to describe it as breaking things down and building things up. So since these reactions, catabolic reactions, you're breaking things down. Anabolic reactions, you're building things up. What if you want those reactions to happen faster? More catalysts or more enzymes, right? So if you are one of those type of people who have lots of enzymes, these reactions happen really fast, then we say you have fast metabolism, okay? And where do you get the directions to make all these enzymes? DNA. So would you say having a fast versus slow metabolism, is that genetic? Maybe, partly, okay. Are there any questions or anything you want to go over for this part? Yeah. Yes, there's definitely been like studies on that, and there's been a lot of correlation that people have found between age and the rate at which you metabolize. And I think you can kind of make that as a conjecture, like that's probably true, because as you get older, your DNA isn't as sprightly as it once used to be. Your cells don't regenerate as fast, your hair doesn't grow as fast, your skin cells don't grow as fast, nothing grows as fast anymore. But it happens to everybody, so if that makes you feel better, like, yeah. All right. I think, oh wait, sorry, one last thing. Functions. So these are actually two examples specifically from digestive systems. In your salivary glands, we have amylase. It breaks down starches into smaller pieces of complex sugars. So if for lecture 5 you do a concept web, and your concept web so far has something like this, which is that first line there, what kind of relationship do salivary glands and amylase have with each other? Secretes. What's another word for secrete? Release. Anything else? Produce. Makes. Which way should the arrow point? There's amylase. Okay. So salivary glands secretes amylase. What does amylase do to starch? Breaks down. Okay. So in your definition, sorry, actually, let's do this. Breaks down starch. I notice starch is under the category of a substrate. So back to your definition of substrate. If you have the textbook definition, awesome. But I'm going to have you add an alternative definition. Substrates, like starch, are going to be broken down by enzymes. And then another example of enzymes doing their job in your stomach. We have an enzyme produced by the gastric glands, pepsin. Breaks down protein so that you can eat your steaks. And we'll break the proteins down into smaller polypeptide chains so that you can use that in an anabolic reaction to build up your muscles and your protein pumps. And then the last portion, I don't know why I wrote wait until lecture. I think you can kind of figure it out. You can use these enzymes in two places, intracellular or extracellular. What does that one mean? Outside of the cell? Inside of the